Okay.
2 minutes, the usual
Stragglers.
It's kind of my week. I've noticed that.
Oh, okay. Do I need to do anything?
Yeah, this week it's not.
I just stand there. Okay, that's all I need to do. I'll stand there.
Yeah, well,
Just camera.
make
Yeah.
yourself virtually in the camera, but you can still wave. Yeah. So thanks everyone here and online. Really pleased to have Rob Newton over to talk about infections and cancer in Uganda, mainly, I guess,
Mainly.
and as missed opportunities for prevention.
Thank you, George. Thanks very much. And thank you for the invitation to come and speak to you today. It's great to be in Bristol. I think I was last here about 20 years ago, actually, 2005.
Down the having a fry up.
Exactly. Yes, in your favourite cafe. That's right. So I'm going to talk about infections and cancer. And I'm going to, I think, relatively quickly walk you through a sort of 30-year career of studying infections and cancer, primarily in sub-Saharan Africa. But by way of introduction, let's see if I can get this to work. There we go. By way of introduction, just to say a few words about the kind of work that I do in sub-Saharan Africa. And throughout my entire career, I've focused primarily on underserved population, people who are not well studied by other groups. And in my case, that's been mainly in sub-Saharan Africa. Of course, there are plenty of places elsewhere in the world where there are underserved populations who require study. And I chose to work in Africa for a variety of reasons, partly because there are underserved populations there, but also because it's the very best place to answer certain key scientific questions. Now, if, as I do, you work on infections and cancer, the cancer-causing infections are all much more common in sub-Saharan Africa than they are here, and the cancers that they cause are also more common. So Africa is an ideal place, and that's what took me there in the very first place. But of course, there are other reasons for going to Africa, and this is another one. This is quite a famous plot published by Bihar and colleagues in 2012, and it basically summarizes human history according to the diversification of mitochondrial DNA. Now, mitochondrial DNA is quite conserved, it's passed down the maternal line, and if you start with human history at the very beginning, you can see an increasing diversity of mitochondrial DNA. But the key point I want to make about genetics in Africa and studying genetics in Africa is the entire breadth of that graph is sub-Saharan Africa. The whole of the rest of the world put together, every other population in the world, is that little pink bar in the very middle. So humanity started in Africa, it's been there longer than anywhere else. It seems obvious, but there is greater genetic diversity on the African continent than anywhere else in the world. It's not my area of study, but if anyone is interested in genetics, Africa is a very, very good place to study it. Okay, the other key feature of the research that I do is it's research for prevention, cancer prevention. And the primary reason for that is throughout most of sub-Saharan Africa, the facilities for diagnosing and treating cancer are scant. I work primarily in Uganda. There's one cancer center. By the time people are diagnosed with cancer, they usually have terminal stage disease. So there's very little you can do for most people who are diagnosed with cancer. And therefore, in terms of bang for your buck, it makes far more sense to focus on cancer prevention than it does on treatment. That's often not where a lot of the NGO money goes. People love to build cancer centers and, you know, donate drugs and so on. But if the expertise is not there to use them properly, you're wasting your money. I focus on cancers that are common in Africa. I'll talk about a couple of those today, but I also focus on cancers that are common everywhere, including in sub-Saharan Africa. And just a sort of a side comment, a lot of the work that we do in Uganda also has a strong focus on local capacity building, with strong international collaborations with the WHO, with the International Agency for Research on Cancer, and so on. I've also in the past had strong collaborations with the National Cancer Institute, although I have to say since the election of Donald Trump, that's coming to an end because the NCI, National Cancer Institute, is under serious threat of financial cuts. The other key feature of my work is the focus is on primary data collection. So I don't particularly work on systematic reviews or secondary data analyses. It's more about collecting primary data. So a key feature of working in sub-Saharan Africa is you may be surprised how little we actually know and how few data there are. And I think that's well illustrated in this slide, which shows the proportion of the populations that are covered either by cancer registration or by vital registration of cause-specific mortality. If you look at Africa, a tiny proportion of the population is covered by cancer registration. So for most of the continent, we have no idea what's going on. And we wouldn't necessarily pick up changes in patterns of cancer unless they were major, as happened when HIV became very prevalent. And I'll come back to that a bit later. But I think of more importance is the fact that none of the continent outside of South Africa is covered by vital registration of cause-specific mortality. So the truth is, we don't really know what people are dying of. And one of the key elements of my work, I'm fortunate enough to work in a well-enumerated cohort, a population in rural Africa, is our emphasis on trying to establish what people are dying of. And I have to say that's much harder than you might think, because Uganda has about 11 or 12 senior pathologists in the entire country. There's no system of assessing cause-specific mortality. So we have to use other approaches to identifying what the major causes of death are. And I think it's pretty critical that you understand that as we go forward, because we're lacking a lot of data. So this is where I work most of the time. I'm based in Uganda, although I have, and I'm going to show you data from Rwanda, which is just south, and a number of other countries on the continent. I'll also show you some data from South Africa. But right now, most of what I'm going to show you comes from Uganda. The Medical Research Council, the UK Medical Research Council, has had a research facility established in Uganda for more than 30 years now. It was actually set up at the request of the president of Uganda when he recognized, when he first took power in 1986, that this, what was then a new disease of AIDS, was really beginning to take off and was very poorly understood on the continent. So the unit was set up to study HIV, but we've moved on and diversified since then. So these are data from the very first study I did. So I went to sub-Saharan Africa because of an interest in HIV-associated cancers. And the reason for the interest in HIV-associated cancers was it was recognized since the very early 80s that AIDS increased the risk of specific cancers. And those cancers were the ones that were either known or thought to be caused by underlying infections, other than HIV itself. So the immune suppression caused by HIV was facilitating the action of other infections. So the very simplistic basis of my PhD, which turned out to be too simplistic, was that if we could identify HIV-associated cancers, we would be identifying cancers that had an underlying infectious cause. That seemed quite an important thing to do at the time. Now, this study was based in Rwanda, and you can see that the numbers of cases of the various tumors are actually relatively low. And the reason for that is this study was curtailed by the genocide in 1994. And I shamelessly fled the country at that time. But we identified 3 major HIV-associated cancers. Kaposi's sarcoma was known about, from studies in the US and Europe. Non-Hodgkin lymphomas were strongly associated, but the new one was a type of ocular cancer. Now, the numbers here are tiny, but we subsequently went on to conduct studies elsewhere in sub-Saharan Africa, in Uganda, South Africa. The South African study is the Johannesburg Cancer Study, which I conducted with an old mate of yours, Freddie Cetus, and is actually the largest study of cancer on the African continent now. We also worked in Malawi and even in Algeria. And basically, these findings were replicated in all these settings with much larger numbers of cases. So I'm going to say a word about each of these groups of tumours, starting with the least common one here, the ocular cancers, and then I'll move on to talk about a non-Hodgkin lymphoma, or particular type of non-Hodgkin lymphoma, and Kaposi sarcoma. So the ocular cancer I'm talking about specifically is this one. It's a squamous cell carcinoma of the conjunctiva. It has a cutaneous cousin, squamous cell carcinoma of the skin, which you see quite commonly in immune suppressed transplant recipients in Europe and the USA. For a while, it was thought to be caused by cutaneous HPV subtypes, but we've subsequently shown that that's not the case. And actually, I have to say, we don't know what the underlying infectious cause of this tumor is, if indeed there is one. So I showed quite early on that the incidence of squamous cell carcinoma of the conjunctiva varied in quite a big way globally, and that the incidence increased with increasing levels of ambient solar ultraviolet radiation. So like the skin cancer, this tumor is clearly associated with sunlight exposure. Cancers have multiple causes. So that was a pretty clear finding. But we then went on to show that there were also quite striking associations with HIV. And this was a piece of work I published in the 90s, summarizing data from 5 studies, 3 of which were my own, which show very clear and very strikingly similar findings of a roughly 10-fold excess risk of this particular eye cancer in HIV-infected individuals. I think the interesting thing about this is these data are now very old, and there have been other studies that have shown the same thing. Many laboratories around the world have looked for an underlying infectious cause, and nobody has identified one. So this remains an open question. If anyone is interested in identifying a new virus, this might be a good place to start looking. So just a comment about a little more detail about where I work in Uganda. It's rural southwestern Uganda. The MRC set up a cohort study there more than 30 years ago. It's not a huge population. It's about 22 or 23,000 people of all ages, from 0 to 100, in 25 adjacent villages. So we recruit everyone who's available at the time within those 25 villages. And that actually is one of the things that makes it quite unique. Most cohorts in Africa, and there are not many, were set up to study HIV and have quite restricted age ranges. Ours doesn't. We have the whole population, which makes it much more interesting if you're trying to study diseases across the entire life course. Now, the cohort, of course, is quite small. It was powered to study HIV, which is very prevalent. It was about 11% in the late 80s, and is a little bit higher now. People are surviving longer on antiretrovirals, so the prevalence has crept up a bit, although the incidence of HIV is declining a little bit. But what we are powered to look at is causes, many of which are very, very prevalent. So a lot of the data I'm going to show you come from this area. A key point, again, about studying African populations is that the demographics of the populations are completely different to what you would find here in the UK or in the USA. So you're probably all familiar with these classic population pyramids. This is Uganda, and it really is a pyramid. 56% of the population are children. There are relatively few older people in the population because survival is quite poor. If you compare that, these are data from the USA, but UK looks pretty similar. the structure of the population is totally different. So of course, that also has an impact on the frequency of diseases that you see. The incidence might be the same, but they're likely to be more common here if they're associated with older age, simply because there are more older people. So that also is something that's important to bear in mind. So one of the first things I did when I went to Uganda to study non-communicable diseases to the MRC unit was I just started counting things. We had no idea what people were dying of. We had no idea what the prevalence of most risk factors for disease was. So I started by introducing a process of what's called verbal autopsy. In other words, when somebody dies in our community, we get to know of it. We interview the relatives in a very structured way using sort of standardized questionnaires to try and identify what the underlying cause of death is likely to have been. Now, it's far from a perfect system. I think we all recognize that, but in this environment, it's all we've got. It's the best we can do. And it has provided us with some useful information over the years. So here, there's data on 1,000 deaths occurring in the cohort over a period of about 10 years. And, oh, I'm sorry, not sure how to go back. And you can see that the most important cause of death, even now with antiretrovirals, remains that combination of HIV and tuberculosis. Roughly 20%, 1 in 5 deaths is caused by HIV, TB. And then there are some very common other infectious diseases, particularly lower respiratory tract infections, pneumonias. Of the non-communicable diseases, an important point, cancer is by far the most important. Here it says 15% of deaths are due to cancer. It's actually slightly higher than that because quite a few of the HIV deaths are associated with Kaposi's sarcoma and non-Hodgkin lymphoma. So in fact, about 1 in 5 deaths is a cancer-related death in this population. And that's interesting because if you look at the proportion of funding that's gone to study NCDs in sub-Saharan Africa, very little of it from the UK has gone to cancer research. Almost all of it's gone to diabetes and cardiovascular disease, and they're relatively less important compared to cancer. Another key point about the cancers we're seeing here is that almost all of them are caused by underlying infections. So we see very little lung cancer, for example, in Uganda because people don't smoke. They can't afford it. Breast cancer and prostate cancer are becoming more common in the urban centres where women are beginning to take control of their reproductive lives and having fewer children. The risks of breast cancer go up. But actually 86% of the Ugandan population is rural. It's not urban at all, and fertility rates remain incredibly high. So then looking at the causes of non-communicable disease in these populations, I'm going to focus particularly on the cancer-causing infections. We've also looked at a range of risk factors for other non-communicable diseases, including lipid levels, HbA1c for diabetes and so on, and they're all relatively less important than the infections. These are the big ones. The top 3 here, Helicobacter pylori, which is a bacterium, not a virus, Epstein-Barr virus and Kaposi's sarcoma-associated herpes virus are more or less ubiquitous in the population. The most common cancer actually is cervical cancer, caused by human papillomavirus, followed by Kaposi's sarcoma, caused by KSHV. And then, of course, you've got liver cancer caused by hepatitis viruses and aflatoxin exposure, and probably other things as well. A range of other cancers that are, yeah.
Have you seen AIDS-defining HPV cancers, or most of your, because most of the stuff we see?
You certainly do. I mean, cervical cancer is by far the most common. It's so overwhelming. The other HPV-associated cancers tend to get lost, but you certainly do see some of the oral cancers that have been associated with HPV. Cancer of the penis used to be very common in Uganda. It's declined a bit with the with the spread of clean water supplies. People can wash properly now, whereas 100 years ago they couldn't. So Sorry.
Do you see cervical cancer in people without AIDS?
Oh, yes.
Yeah.
Oh, yeah, yeah.
Can-
Very much so. I mean, the risks are increased in people with HIV, but the majority of people with cervical cancer will not have HIV as well. That isn't true with Kaposi sarcoma. KS was always common in Uganda, even before the HIV epidemic. I'll come back to that later, but it still remains a very strongly HIV-associated disease. But I think a couple of key points about this. The first is I want to emphasize again that top of the list is Helicobacter pylori. This is a sort of forgotten infection in my world. Stomach cancer has been declining in richer countries for many, many years because H. pylori is declining in prevalence for reasons we don't fully understand. But here, it remains a really important infection. And remember, it doesn't just cause stomach cancer. Actually, there are more deaths associated with upper GI bleeds, with duodenal and gastric ulcers, than with stomach cancer. So this is a major, major problem about which we are doing nothing. That's potentially curable with antibiotics, but there's almost no vaccine research in H. pylori anywhere in the world. China looked at it a few years ago, but have abandoned it. So it really is a sort of forgotten infection. EBV and KSHV are related, gamma herpes viruses, and I'll say a bit more about all of those a little bit later on. Where I work, Hep B is not that common, but elsewhere in Uganda, the prevalence can be higher than 20 or even 25%. And we do not understand why the prevalence varies that much between geographically quite proximate regions. We have some preliminary data that suggests that schistosomiasis infection, which is common around the Great Lakes regions, is associated with increased viral loads of hepatitis B, and that would facilitate spread and pathogenicity. But that's very preliminary data. But if you look at the geography of Hep B in Uganda, actually it is highest around the Great Lakes regions where schisto is most common. So that's something for further exploration. Hep C, actually very rare. Interestingly, when we tested the cohort a number of years ago, we tested about 8,000 adults. We found 13 cases of hepatitis C. About half of them died relatively quickly of liver disease, and the other half we cured. So my great claim to fame is that I've eradicated hepatitis C from our cohort population, all 6 cases or however many I treated at the time. I'm going to say a word about this tumor. This is African Burkitt lymphoma. This is an important tumor that was first described by Dennis Burkitt in the late 50s, 1958, and it was the first human cancer to be associated with an underlying infection, the Epstein-Barr virus. So it actually has a really critical place in tumor oncology in our understanding of the causes of cancer globally. Interestingly also, it was one of the first tumors to be successfully treated with combination chemotherapy. So it really has, and this all happened in Uganda. This all happened at the Uganda Cancer Institute. So another good reason for not neglecting sub-Saharan Africa if you're interested in cancer research. So what were my contributions in relation to Burkitt lymphoma? I'm showing you these data because it emphasizes the importance of infections and also of the interaction, the complex way in which co-infections work together. So this is a little bit complicated, just bear with me. We measured levels of Epstein-Barr virus and of malaria in a case control study. And I've divided those individuals up into people who have low levels of EBV and high levels of EBV, low levels of malaria and high levels of malaria. Now, if you look at the risks on the far side, they're actually not odds ratios, they're floating absolute risks, but don't worry about that. You can see that the excess risks are all in those individuals who have high levels of both infections. It is not sufficient to have a high level against just one of them. I replicated these findings in Malawi, and believe it or not, it was the first evidence within individuals that malaria could act as a cofactor for cancer in relation to this tumor. And what's interesting, I'll come back to malaria again, is EBV as a gamma herpes virus, is not the only gamma herpes virus that's affected by malaria. We see the same for Kaposi's sarcoma virus. So I'll come back to that a little bit later on. So Kaposi's sarcoma, this is the big one, the second most common cancer in Uganda. It really became famous in the early 1980s because this was one of the diseases, together with pneumocystis pneumonia, that heralded the onset of the HIV epidemic in Los Angeles and New York amongst men who have sex with men. But it's always been common in Africa. In fact, in Uganda, it represented about 9% of all tumors in men, even before the HIV epidemic. So that's on a par with colorectal cancer in the UK. That's the sort of levels we were talking about. And it exists in different forms. Traditionally, we saw the lesions on the lower limbs, and it's quite a severe case here, obviously, primarily in men. But with the spread of HIV, we started seeing these much more aggressive forms of Kaposi's sarcoma that would cover the whole body. And there are also systemic lesions, internal lesions in the gut and the lungs and so on. So it remains a very, very important disease. The lady I photograph on the right-hand side is my colleague Denise Whitby, with whom I've done a lot of this work. So what did we show? Well, one of the first things we showed, just looking at Kaposi's sarcoma in sub-Saharan Africa. Now, this is a tumor that outside of a handful of high-risk populations, primarily gay men, doesn't really exist anywhere in the world outside of sub-Saharan Africa. And even within sub-Saharan Africa, we see tremendous variation in incidence. In Uganda, as I say, it represented 9% of all cancers. Elsewhere in sub-Saharan Africa, it was very much rarer. This piece of work was done many years ago, and actually was Dennis Burkitt's last publication. It was published after his death. So I have the rare privilege of having co-authored a paper with Dennis Burkitt, who's quite an icon in the world of cancer research. So what we went on to do when KSHV was discovered was compare KS incidence in Africa, and that's an updated map on the right-hand side at the top, with the prevalence of KSHV across Africa. And you can see these very, very striking associations where KSHV is common, KS is common. That's hardly surprising. It is now established as the underlying cause. And it was established as the underlying cause with the usual approaches that we use in epidemiology. This was data from a case control study that I did with my friend Freddie Cetus. We looked at, he'd been recruiting cancer cases in South Africa for many, many years. We looked for the prevalence of KSHV in people with all these different cancer types. And you can see that the one that stands out is Kaposi's sarcoma. Huge excess risks of KS associated with KSHV. So it's these kind of simple case control studies that really firmly established the causality of KSHV. We then went on to do prospective studies. I did two, one here in the UK, in the context of clinical trials against AIDS, and this one from sub-Saharan Africa. And what we showed was, you can see the red line there, that's the antibody levels against KSHV in individuals years prior to diagnosis. So on the right-hand side is where the KS was diagnosed. On the left-hand side, we're up to 6 years prior to diagnosis, and the antibody titers were much higher. We found the same in the UK. So this is just classic epidemiology and longitudinal studies, very, very clear associations. We were also able to show in very simple studies that antibody titers against KSHV were important as a determinant of transmission of KSHV. In this case, we looked at transmission from a mother to a child. So if you look in very young children, the prevalence of KSHV is very strongly associated with the mother's status. If the mother has KSHV, she's positive against KSHV, you see a much, much higher prevalence in the children. And again, all these studies have been replicated on countless occasions and on much larger numbers. So what about KSHV in our cohort? in Uganda. Well, I'm putting these data up because it highlights a problem we see with all of the cancer-causing infections in this population. The first point to make is it's highly prevalent. In this case, by age 10 or 20, it's more or less ubiquitous in this population. The second point I want to make is aged infection matters. We know from other studies that the younger you are when you're infected with KSHV and HPV and hepatitis B has an impact on the subsequent risk of cancer. The younger you are when you're infected, the higher the risk. We've got very early age of infection here. And the third point I want to make is illustrated in the panels on the right-hand side, which shows the distribution of antibody titers in two populations, one from the USA on the top panel and one from our population in Uganda on the bottom panel. And you can see that the distribution of antibody titers is totally different. We have much, much higher antibody titers in the African population than we do in the US population. And again, that matters. High antibody titers against this virus and other cancer-causing viruses reflects the subsequent risk of cancer. High titers, increased risk. So we've got a sort of trio of problems here. High prevalence, early age of infection, and very high antibody titers. And that's reflected in subsequent risk of the cancer. I'll come back to malaria again. Yeah, another gamma herpes virus, KSHV. We found that both in adults and in children, malaria is associated with an increased prevalence of detectable antibodies against KSHV. We now know that malaria impacts on lytic replication of the virus. In other words, how much the virus is replicating within individuals. And that matters both in terms of risk of transmission to somebody else, but also in terms of subsequent risk of cancer. So malaria is bad news in this population, and it may be that one of the most effective ways to reduce the burden of KS in sub-Saharan Africa is to vaccinate against malaria. I'm now working with colleagues at the Jenner Institute in Oxford who have produced by far the most effective malaria vaccine, which they're rolling out, and hopefully we'll know in the next couple of months whether the vaccination has any impact on levels of KSHV within individuals. But it's not just about lytic replication. In a more recent study, we've also shown that malaria has an impact on susceptibility to KSHV. So we looked at young children between the age of 1 and 2 years, the prevalence of KSHV increased much more rapidly in the children who were also diagnosed with malaria at some point during that period. So that's the solid line that's higher up on that graph. The dotted line at the bottom is the kids who weren't diagnosed with malaria at any stage during the study. So increased susceptibility against KSHV and increased risk of cancer associated and transmission associated with malaria. So that deadly combination of co-infection, once again, matters. We've seen it with HIV and the infections that are associated with cancers in AIDS, and we see it with malaria as well. And of course, you've got to remember in Uganda, malaria is ubiquitous. It's a holoendemic infection. It's very difficult not to be exposed to malaria at some point in your life. So its potential population level impact could be huge. Okay, we've looked at genetics. The population on the study has undergone a GWAS analysis. I took advantage of that and looked for genetic associations with KSHV. My focus has always been on environmental factors that impact on spread of infection. But we did look at the genetic factors, and to be honest, we didn't find much. There are a few modest hits in the HLA regions, but as is often the case with GWAS analyses, the relative risks were very small, less than 1.5. So not much to say in relation to genetics there. It seems to me, on the basis of data we've collected over the years, that it's environmental factors that are impacting more on the prevalence of this infection in sub-Saharan Africa than genetic factors. And certainly, if you look at Africans who've migrated off the African continent to, for example, America, within a generation, they've lost KSHV completely. So there is certainly something on the continent that is facilitating continued spread of the virus. More recently, I've become interested not just in antibody titers against KSHV, but in viral loads. And I'm just putting this up by way of illustration. If you look in people across the life course, these data are based on samples from 1,000 people in our cohort of all ages. You can see that the prevalence of detectable KSHV in saliva, which is the blue line, that's how it's transmitted, varies hugely with age. It also varies hugely with sex. That isn't evident on this graph, but it looks like that perhaps the major transmitters of KSHV are adolescent males. They have much higher levels of infection in their saliva than do females. And we find the same for detectable KSHV in blood. Now, this matters because saliva is associated with transmission, blood is associated with pathogenesis to disease. So it is relevant, and it's also relevant that it's predominantly a male problem, because outside of the context of HIV, Kaposi's sarcoma is predominantly a male problem. So there's something about being male that is facilitating pathogenesis to the cancer in this case. Again, no idea what, but I think that's quite interesting. Another point to make about viral, oh, and this is levels of malaria parasitemia, and you can see that it sort of mirrors what we're finding for the viral loads in saliva and in blood. But the other interesting thing we found was that there was absolutely no correlation within individuals between having detectable virus in blood and saliva. Having one didn't make you more likely to have the other. And that suggests that the immune control of KSHV in the two different compartments, the oral cavity and systemically, is different. And that's something we don't understand at all. It's never really been studied. So we're doing quite a lot of work now to look at parameters within the oral cavity that might be associated with presence of the virus, like dental health, for example, and various other factors, in an effort to understand why it's shed in some individuals and not others, obviously with a focus on trying to prevent transmission in saliva. We've also looked at immune control of KSHV in blood. This is a complex slide, and I'm not going to spend too much time on it. But in essence, what we did was sequence every protein on the exterior surface of KSHV, every single thing that could be presented to the human immune system. And using ELISpot assays, we tried to identify what provoked an immune response. This virus is interesting because compared to EBV or CMV, two other herpes viruses, which are in the control panels on the right-hand side, KSHV provokes a minimal immune response, and there's massive heterogeneity in that response. There's no one antigen that is provoking a bigger immune response than any other. And that may be important in terms of vaccination in the future, if anyone can be bothered to produce a vaccine. I'll say a comment about that in a moment. Because it may not matter what you target your vaccine against. If you can provoke an immune response against any one of these things, it might actually have a big impact. The problem with developing vaccines against KSHV is that there's almost no commercial interest in doing so, because the population at risk is almost entirely African, and Africans can't afford to pay for vaccines. So the motivation to develop a KSHV vaccine, even given the fact that it's likely to be very successful, is low. And we've had absolutely no success so far in persuading anybody to develop a vaccine. I mean, we organized a meeting at the National Cancer Institute a couple of years ago, 3 years ago, to sort of try and provoke some interest, and we got nowhere. So that's really quite disappointing, I think. So what next for KSHV? Well, we're looking for other paths to interventions. I'm interested, as I've said, in transmission and whether we can impact on transmission by impacting on shedding in saliva. I'm interested in controlling pathogenesis. That means controlling lytic replication of the virus in blood, viral loads in blood. And the long-term goal of all of that research, of course, is interventions. And there are various points in the pathway between exposure to infection to onward transmission or development of disease where one might be able to intervene in the future, but we still have an awful lot of work to do. I'm going to make a quick comment now about a tumour that we don't think is associated with an infection, but I'm raising it just because it's a piece of work that I'm really quite proud of. We set up a treatment study of this childhood cancer retinoblastoma in Uganda. One of the reasons for doing that, so it's been evident for decades that retinoblastoma is one of the only childhood cancers that does seem to vary in incidence around the world. It's more common in poor countries than it is in rich countries. There's roughly a threefold difference in incidence. I mean, it was quite challenging to look at because it's not that common a cancer. So small variations in the number of cases can impact on your guesstimate of incidence, but it does seem to be real. So more than 20 years ago, I set up a case control study of retinoblastoma in Uganda to try and identify underlying causes, discovered absolutely nothing. In retrospect, we were asking completely the wrong questions. But what became evident very quickly is that every single one of these kids was dying. Nobody dies from retinoblastoma in Britain. Almost, I think in the last 5 years, there've been maybe 1 or possibly 2 deaths from retinoblastoma in the UK, and both occurred in immigrants who were diagnosed very, very late. So it's generally picked up early here and treated very successfully. So it was a bit of a blow to find in Uganda that every single kid was dying for such a treatable tumor. So we introduced a program of neoadjuvant chemotherapy. In other words, these tumors present so late that you cannot surgically remove the whole thing. So we gave these kids cheap, widely available chemotherapy drugs that we brought in illegally from Kenya. And the aim was to debulk the tumor prior to surgery. So you had some hope of getting the whole damn thing out. And it was spectacularly successful. Survival has increased dramatically. We now save more than 75% of these children. 15 years of follow-up will be published this year. We've saved the lives of more than 1,000 children. So that's one area where focusing on treatment of cancer can have tremendous success. And it really isn't expensive. It costs us about $600 to cure a child from retinoblastoma. We would be spending about $600,000 in the Western populations of the world to cure this tumor, but you can do it much, much more easily. So I just put that up because it's my vanity project. In terms of infections and cancer, again, I'm working into the future. I've talked quite a bit about KSHV, susceptibility, reducing viral loads, and so on. But there are other infections that remain very, very important. HPV, as you know, has a very safe, very effective vaccine against it. And that vaccine works very well in sub-Saharan Africa, even with a single dose. You get 3 doses in this country. The problem is people don't get it. Vaccine coverage for all vaccines, including some of the childhood vaccines, is so critical for preventing infectious diseases, doesn't reach more than 60%, 55, 60% in our cohort for any of the common vaccines that are given. The primary reason for that is that 40 to 50% of kids are not born in health facilities and never interact with health facilities. So they never have opportunity to be given the vaccines. And that really is a huge issue and a huge failure of prevention. I mean, there's lots of research going on vaccines in Africa and people worrying about whether co-infections reduce the efficacy of vaccines and so on. By far, the most important thing is vaccine coverage. If you're only vaccinating half your population, these diseases will never go away. So for HPV, I've been focusing on vaccine coverage. We know from our research in the cohort that only 40% of girls have even a single dose of the vaccine, and about 2% get all 3 doses. So at those levels, and bear in mind, we're not vaccinating boys, so you're covering a tiny proportion of your infected, potentially infected population. Our chances of really impacting in the future on cervical cancer incidence are much diminished. And so I think that's a major area of focus. For HBV, we have the same problem. That's a vaccine that's given to boys and girls in infancy, but only half the kids are actually getting it. And that's why we still have a very high prevalence of hepatitis B, even in young children in Uganda and elsewhere in sub-Saharan Africa. The other issues for HBV, and I think this is important again to remember, is there are more people in the world chronically infected with hepatitis B than with HIV. It remains a major global infection causing liver cancer and other types of liver disease. And actually, some of the issues for hepatitis B are almost the same as for HIV. If you can reduce viremia with a drug, you reduce the risk of onward transmission, and you reduce the risk almost to zero of pathogenesis of the disease, exactly the same way that antiretrovirals do for HIV. And actually, the drug we use to treat Hep B is one of those antiretrovirals, tenofovir. So it's cheap, it's widely available, but it isn't yet being widely used in the treatment of Hep B. And even identifying who's got chronic Hep B in your population is challenging, because there isn't sort of routine screening in most of the populations in which I work. For H. pylori, the bacterium, I've already mentioned the issue of vaccination and the fact that nobody is developing a vaccine. The other problem we have is we can't even treat it effectively because there are massive levels of drug resistance. I mean, in people over the age of 40, symptoms from Helicobacter pylori, gastritis and so on are almost ubiquitous. And we do try to treat it, but the antibiotics don't work. There's far too much drug resistance in our population. So we don't even know how to effectively treat H. pylori. So that, again, represents a major problem. And I suppose the take-home message, if you like, is these problems are solvable if you spend the time to do it and you spend the money to do it. But as things stand, the investment in cancer research in sub-Saharan Africa is minimal. So we're not making as rapid progress as we should. I'm going to finish by making a comment about another area of interest of mine, which is focused on the non-specific effects of infections and inflammation. And that's the impact they have on hematological malignancies. Now, it's long been said, inaccurately in my view, that the diseases such as non-Hodgkin lymphoma are less common in sub-Saharan Africa than in the Western world. And that makes no sense to me, because the things we know that are associated with non-Hodgkin lymphoma, infections, inflammation, and so on, are all much more common in sub-Saharan Africa. What seems much more likely to me is that we're simply not diagnosing them. So many of these hematological malignancies present either with a swollen lymph node or just with a fever. And in sub-Saharan Africa, those things will be treated as something else without ever having a proper diagnosis. And if the person has got non-Hodgkin lymphoma, they will die long before you ever know it. But in recent years, we've developed approaches for looking at pre-malignant hematological conditions. Many of them have preclinical manifestations. And the advantage of those is you can screen for them. So for the first time, we can actually properly assess the prevalences of these pre-malignancies in populations in sub-Saharan Africa and in the global north. So I've looked at monoclonal B-cell lymphocytosis. That's generally in the UK, that's a precursor to chronic lymphocytic leukemia. But CLL is defined on the basis of the number of circulating malignant cells in blood. If you're below a certain level, you have MBL. If you're above a certain level, you start getting clinical manifestations and you have CLL. But you can measure MBL in screened populations. I've also looked at MGUS, monoclonal gammopathy of undetermined significance, which is associated with multiple myeloma. I'm not going to present any data on that because it's not ready yet. And at age-related clonal hematopoiesis, which I'll say a word about in a moment. So in relation to B-cell lymphocytosis, we looked in the general population cohort. And what we found actually was total levels of MBL were quite a lot higher in our African population. This was against many people's expectation than we found in the UK. But what was particularly interesting, I think, also, is that the phenotypes of that MBL were completely different. In the UK, it would all be CLL related, chronic lymphocytic leukemia related. In Uganda, we didn't find any CLL. It was marginal zone lymphomas. So the phenotypes of what we were finding were completely different as well. Now, that has quite significant implications for our study of hematological malignancies in Africa, because these things are not easy to diagnose. And the way we define them is based on years of work on defining tumor markers, genetic factors associated with the cancers. If the phenotypes in Africa are completely different, the approaches we're using here may not work because the tumors are manifesting in a different way. They have very different phenotypes. So that's something else to bear in mind. We did look where we could at why the MBL might be more common in the Ugandan population. There are a number of well-established SNPs, genetic markers that are associated with increased risk of CLL in Britain. And we looked using our GVAS data at the prevalence of those genetic markers in the UK compared to Uganda, and the prevalence was much lower in Uganda than it was in the UK, which probably has something to do with why we didn't find any CLL phenotype. Something else was going on instead. So this is all very preliminary data based on quite small numbers, but is nonetheless quite interesting, I think. Now, a word about age-related clonal hematopoiesis. I don't know if any of you know anything about it. Basically, what happens, your circulating blood cells are made very largely in adult life by stem cells in your bone marrow. As we get older, we become more clonal. Those stem cells pick up mutations, and sometimes those mutations confer a selective advantage to that cell lineage, and it begins to take over within your bone marrow. And that's bad news, because if you become more clonal, it's associated with increased risk of hematological malignancies and of cardiovascular diseases because you become less effective at manufacturing blood cells that you need. In Western countries, the prevalence of age-related clonal hematopoiesis is around 10% in people over the age of 70. Now, I decided to look in Uganda primarily because we were finding an incredibly high prevalence of anemia in older people particularly. And anemia in older people in Africa has been very badly studied. The focus has always been on infants who have a lot of anemia because of malaria and on pregnant women. But in fact, the bulk of anemia that occurs in the population is in older people, and we have no idea why. You can only explain a small proportion of it on the basis of parasite infections and so on, the things that we know about. So I looked at age-related clonal hematopoiesis, and indeed I found it was associated with anemia in the Ugandan population, but also that the prevalence was substantially higher, 25% in 40 to 60-year-olds, and nearly half of people over the age of 60, compared to that 10% in 70-plus-year-olds in Western populations. And that probably has quite important implications for subsequent risk of disease, both cardiovascular disease and hematological malignancies, as African populations begin to age more than they currently do. So again, that's another huge area of interest. We don't know what factors are impacting on these things, but they're probably infectious, maybe not a specific infection, maybe just the incredible burden of antigenic exposure that people have living in populations with high prevalence of all sorts of different infections. So I think I will stop there, but I'm very happy to take questions if anyone wants to take the pot shot.
That's great, real old school proper epidemiology. We're an epidemiology unit, very rarely have talks, which are proper epidemiology. So
You can't do anything else in the context of sub-Saharan Africa.
That was wonderful. Questions?
Please.
Can I just ask very simply, like what proportion of the variants of Kaposi sarcoma can we explain by, say, under 5 prevalence of KSH2? Like, is that like 70% of the variants, or?
No, I think it's very, very difficult. I mean, the biggest explanator, if you like, of KS is co-infection with HIV in an already infected individual, to someone who's already got KSHB. So if your background prevalence of KSHB is close to 100%, and then you get HIV, the incidence of KS goes up markedly. So in the 80s and 90s, the incidence of KS increased 30-fold in Uganda. It's since come down because of the more widespread use of antiretrovirals, but it remains the second most common cancer because not everyone's getting antiretrovirals. All people are getting them too late and have already got Kaposi sarcoma. Interestingly, from a clinical point of view, we're now seeing different forms of Kaposi sarcoma. So it always existed in the HIV negatives, primarily men, relatively low levels, but more common than anywhere else. Then, of course, with HIV and immune suppression, we see a hell of a lot of it still. But now we're also seeing it in people who've been on long-term antiretrovirals and are virally suppressed against HIV. So they've got high CD4 counts, they're not sick from their HIV, but they're developing KS. And we don't know why. It's probably something to do with immune exhaustion of the mechanisms that we use to control KSHV in our bodies. But it really hasn't been well studied. The difficulty with that particular clinical form of KS is it's almost impossible to treat. If someone with acute AIDS and a low CD4 count has KS, you bang in the antiretrovirals and the KS will disappear. That doesn't work for the older people, and they die. So it's a problem that's become manifest as more and more people have been on antiretrovirals for a long period of time.
You looked at any sort of assayable early on, if you like, indicator of differences in response to H. pylori, for example, by age of infection, because this sort of classic work by Zina Sussa in the 1960s basically suggested that that very early H. pylori was associated with stomach cancer risk, and then basically it was as age of infection was delayed, when you were starting getting H. pylori, you weren't getting H. pylori around birth very early on. Then it became, then you see gastric ulcer, and then
duodenal ulcer later on.
And then everything disappears.
Yeah.
In that sort of way.
We haven't been able to look at that in this population yet. I mean, the studies that we've done so far have been serological, and the primary things we've learned is that infection occurs very young. By age 10, it's ubiquitous. Antibody titers are very high, which is bad news. And the third thing that we learned, which is also bad news, is that nearly 100% of infected individuals in Uganda have really high-risk strains of H. pylori, the so-called CAG-A strains, which are highly inflammatory. So it's sort of bad news on all fronts. We haven't yet been able to look at the impact of aged infection, but if all the infection is occurring before 10, I'm not sure how easy that would that would be to do.
But stomach cancer rates are surprisingly not sky high, are
They're not sky high, although it's not easy to measure. Most of there is a cancer registry in Uganda based in Kampala, the capital city. As I mentioned, most people don't live in urban centres, so patterns of cancer in Kampala are quite different to what you see in rural areas. And in rural areas, cancers are diagnosed late, and anything that's occurring sort of between here and here is diagnosed as an abdominal cancer, uncertain cause. So the true incidence of gastric cancer in this population is actually very difficult to ascertain. Very problematic.
Other questions? No. Well, Rob's staying around.
Yes, thank you.
Meetings room, so if you'd like to have a chat about anything.
Yeah, please,
Yeah.
please feel free to stick around.
Thanks again.
Thank you very much.
So I'll go and get you what sort of sandwich would you like? I should get you a sandwich.
Tuna?
Tuna sandwich.
I've got loads of questions to ask.
Yeah, well, let's take a seat and
So many, very interesting, super interesting work.
can you just remind me what you do?
I'm an infectious disease doctor. And